important for the clinic. And thank you very much. I'm now going to hand over to Sarah. Thank you.
That's great. Thank you very much. So I'm very pleased to be here tonight. And so this is the final slot in our session about do we know too much about cancer? And I want to talk about why knowing too much is not enough. And I want to hopefully give you an understanding of, despite the increasing wealth of information we know about cancer biology, actually translating that knowledge into measurable patient impact is actually really quite tricky. So this is a cancer multidisciplinary team meeting, and in hospitals up and down the country, this is how many patients' care plans are decided. And it involves putting together in one room a huge number of different professional specialties. So we have surgeons, we have oncologists, we have pathologists, we have nurse specialists, and together sit down and look at the available information we have about a patient and try and decide the best course of action. For what it's worth, it's one of the most expensive meetings that the NHS runs by the time you accommodate the hourly rate of the people in this meeting. And because of the complexity of cancer as a disease, and because of the complexity of the professional opinions you have in the room, many of the conversations wind up looking a bit like this. So there is a very serious cognitive burden to making good decisions about cancer patient treatments. This is a Cambridge Institute. This is where we work, and it is an absolutely amazing scientific research institute. And I'm privileged to work in this place alongside my colleagues who are absolutely pushing the boundaries of what is possible scientifically to get a better understanding of cancer disease. And some of our answers to the questions that you see in the previous slide look like this. And this is just a selection of some of our publications and some of our research outputs that we've done this year alone. And you will see that it's actually quite a long way from our answers that we produce in the lab to being able to make clinically meaningful changes, right? And there is a long way to go between taking what we do with all the data we generate and then being able to change a clinical decision in the real world. So how do we get from all this huge tsunami of information that we generate in the lab, in the research setting, into something that can actually make a difference to patient outcomes? And so one of the problems is that whilst we have an ever-increasing amount of data, if we want our methods, particularly around big data and AI and those kind of data-driven technologies, to reach escape velocity and get out of the lab into the clinic, we actually need real-world data. And I've given you an example here for a paper that came out of the Cambridge Institute actually quite some years ago now. And it's a method that uses a huge amount of data to be able to predict for patients with breast cancer whether or not they will respond to treatment or not. And in order to do that, this is kind of what we use as ingredients. We had incredibly sophisticated computational environments, and we have a whole squad of people whose job it is to keep our high-performance compute environment up and running and kind of meet our every computational need. We also have a huge amount of immune information, some of you have heard about in the previous talks. We have information about lymphocyte density, cytotoxic activity, lots and lots of really rich immune data for all of the patients in this study. We also have some sequencing data. We have an increasing library of sequencing technologies that we can use to say ever-increasing amounts of information about individual cancer genomes. We have whole genome sequencing, we have cell-free DNA, we have TMB, we have copy number signatures. There's a huge amount of information we can extract from tissue about the genomes of these cancers. We have lots of fancy imaging techniques, and importantly, we have extensively curated medical records. So we have teams of people who are pulling together the clinical histories of these patients, making sure they are neat and tidy and ready for further use. And this takes years. You know, your average scientific project is anything between 2 and 5 years. So we have the time, we have the team, and we have the equipment to be able to put together methods such as this one that we wrote up, which is telling us, giving us the ability to tell us how patients will respond to treatment before we treat them. Not too far away, in the Department of Oncology in Addenbrooke's, we have very little access to any of those technologies and to any of that data. We don't have a high-performance compute environment that's readily accessible in the clinic. We don't have immune data, standard of care data. We are in Addenbrooke's, we have a fairly sophisticated electronic patient record. That is not the case in many hospitals. So there's a huge disparity across the NHS about a clinician's ability to access data through an electronic record. We have some panel sequencing that comes through these organisations that the NHS runs called the Genomics Laboratory Hub. The availability of a test is decided through the NHS test directory, and in order to get a genomics test into the test directory, you need years of evidence to make sure that this is actually a clinically useful and affordable test that will make a difference in the clinic. We also have some access to medical imaging standard, but more importantly, we have clinical timescale pressure. We don't have years to gather this information and deliver a clinically meaningful change. So we're doing it under a very, very different timescale compared to the researchers who are generating these methods in the lab. So if you want methods such as this big data method, these AI methods, to make it into the real world, you need real world data. And there are two ways of doing that. You can either build your methods around data you know will be in the clinic, which is actually really quite basic when you compare it with the richness of data we can generate in the lab. Or you need to spend many years taking research-grade assays and developing them and validating them for clinical use and then convincing the NHS that they are a good use of money. So that's your first point, getting the data that builds all these incredibly sophisticated methods into the real world. Then you have to know your data. And you need to make sure that the people building these methods are actually competent and working with the right people to make sensible decisions about data utility and what you do with the methods. So this is an example of where that did not happen. So some colleagues at the university had a look at some of the big data machine learning methods that were built during the COVID pandemic. So during the COVID pandemic, there was a huge amount of data generated. Lots and lots of CT scans, chest CT scans, lots and lots of chest x-rays as well. And generally, in many cases, these went alongside a PCR test, where we had a lab test that was saying whether or not a patient had COVID-19 or not. And so lots and lots of data scientists get very excited by large data sets. And many, many people decided, let's see if we can build a machine learning method that will tell from a chest x-ray or from a CT scan whether or not a patient has COVID-19, and maybe the ability to decide at the point of taking a scan whether a patient was likely to need to be admitted to intensive care or whether they were likely to have a minor case of COVID-19. And to cut a long story short, there are around 2,000 studies in the literature which are looking at trying to use big data methods for this kind of COVID-19 work. Except not a single one of them is actually fit for clinical use. And this is mainly because of the biases that the researchers were introducing into the models as they built them, and the unsuitability of the data sets they were using for actual clinical use. And I think two favorite outcomes of this was that one of the common data sets that researchers are using as a negative data set for patients that didn't have COVID-19 was actually a pediatric data set. So a lot of the models could happily tell you the difference between adults and children, but not necessarily COVID-19 cases or not. And another flaw in some of the data sets that people were using gave you a set of models which was able to discriminate between patients lying down, who were generally more ill because they were lying down and unable to have a chest x-ray standing up, and patients who had their medical imaging taken standing up. So there's a real need for people who are building big data models, for people who are engaged in AI research, to really actually understand the clinical problems that need solving in the real world. And it's only by that team sport effort of making sure those people are working and talking together that you can get sensible answers. And so, if you have real-world data, assuming you've got a beautiful real-world data set, and assuming you've built a really functional team of big data scientists and clinicians and basic researchers, congratulations, you've gotten to the starting line. That is only the first marker on your journey to the clinic. And so we are separated, the Cambridge Institute is 250 metres away from the site of the new Cambridge Cancer Research Hospital, which is up on the top, and the doors should be opening in 2029. But it takes at least 3 to 5 years to get a method from the lab into clinical practice. And that is if everything is working really, really well. And I think if you look at the analogy of using an interventional medicinal product, so a new drug, there is a very well-worn path about how you can generate evidence for a clinical intervention. For digital methods, for big data methods, we are not yet there in our experience as a community of doing that reliably and at scale. And so this is just an example of some of the problems that you need to solve if you have an excellent method in the lab before it gets anywhere near a patient. So for example, I've already alluded to the fact that the NHS is not awash with high-performance compute. So that is a problem. We need to understand the regulatory environment. So software as a medical device is regulated, and you will have to have any algorithm for clinical use certified as a medical device. You need to understand the information governance behind this. Who's looking at your data? Who can see it? And how is it working? And perhaps most importantly, you need to understand how any method integrates into a clinical data flow. You know, is it acceptable for clinicians to use it? And not just clinicians in major medical centres such as Cambridge and London and Stanford, but for clinicians up and down the country, across the NHS, across the health services. Can they access these tools? Can they integrate them into their clinical workflow? So I'm just going to finish with some examples that we are doing currently in Cambridge, and we are trying to build better tools for data integration. It's worth noting that on the previous slide, none of this work is paid for by medical research charities. None of it is paid for by the NHS. So there is a bit of a funding gap in trying to address all these real-world problems of taking exciting big data technologies and moving them into the real world. But we are partnering, in this case, this is a commercial collaboration we have with GE Healthcare, which is an attempt to aggregate all the data you saw in that first slide of MDT, where we have the medical imaging, we have the pathology, we have all the clinical history, into a more standardized and streamlined format for easier digestibility to try and reduce that cognitive burden. And actually, AI is really quite useful at this. It's quite good at taking vast amounts of text data and generating summaries. And we've worked closely with them to understand what clinicians want to see, when they want to see it, and how they want it presented to them in a way that's easy to digest and kind of reduces the intensity of trying to assemble and trying to aggregate in your head these large volumes of clinical data. And you can see here we have molecular profiles as well. We've got lab results and so on and so forth. But I wanted to finish with an example that is a Cambridge success story and a really nice example of where this has gone well and been done correctly. This is a method called Predict Breast Cancer. There's also a post-rate version as well. And it ticks all of the boxes. It's used millions of times already, and it's designed to be used in conjunction between a patient and their clinician. It's passed all the regulatory frameworks, it's CE marked. And you will see here, it's actually, this is the real interface. It's a web interface. And it uses actually a really quite a small number of fields. If you look at all of the data we know about a patient, we don't actually need that much. And that alludes to Claire's point. One of the challenges is finding that minimal data set you actually need to make a sensible decision. So you have this small set of data, and a patient will sit down with their clinician and fill in their necessary values. And it's got a really nice simple output, which in this case is a graph showing that this patient, for their particular profile, should they choose, in this case, surgery, 87% of patients like them will still be alive in 10 years. So it's a really nice, easy to digest metric based on data analysis and data integration techniques. So I'm going to finish there, but hopefully I will have convinced you that even as we push the envelope and generate ever increasing amounts of data about cancer, it is still nowhere near enough to make a difference to patient outcomes. And I think we are going to take some questions. Is that right? Oh, okay, yes. Well, we're open to any questions. Yeah, that's good. Oh, okay, that's great. I've got a minute. Okay, no worries.
Can I ask you a question about, did you mention you had some separate organisation and hospital that had very advanced analytics and a team that were making decisions on a subset of patients? Is that true?
So we can do that for research data sets that we have, and that's in the lab.
And you can apply that to a subset of patients on a clinical trial?
Yeah, we could do. Yes, you could write a clinical trial and do that. Yes, that's correct.
Yeah. And what are you allowed to do within that clinical trial?
Right.
Are you allowed to access non-standard of care medications?
Right.
What's the limitation of what
Right. So that very much depends on the clinical trial design, and as part of the clinical trial design, when you're writing that protocol, you would think about all the information you want, and that absolutely would include all the other medication a patient might be taking. So that is limited by what you think about when you're setting up the trial. And then as part of that trial setup process, you have to go through the ethical approval process, and the ethic committee will say, yes, that's a sensible use of that data, yes, you can have it.
But would that include drugs that were, say, not for ovarian cancer, but for another cancer that may
Oh, so you mean for the clinical trial itself? Would you give? Yes, absolutely. That happens. Yes, that happens in clinical trials all the time. There'll be a product that's technically for another cancer type, but can be used in a clinical trial for a different type of cancer. Mm-hmm.
But we're talking about this very developed sort of clinic situation in London or Cambridge, for example, where they have very extensive sort of electronic medical records.
Yeah.
Are we looking at bringing them to more developing or low and middle income countries, whereas the doctors may not even be aware,
Right.
or even the system, the medical records are on paper and be lost all the time, and it takes a lot of training to get these sort of things done. Like, is there any initiative to take those into those countries where,
Right.
say, you don't even have steady electricity or internet?
Right. So it's a massive problem, and I'm not aware of any systematic level effort where that's been undertaken. I mean, you guys might know more.
So I think that's a great question, because if you look at the way pharma develops medicines for the United States and for high-income countries that can afford the burden of very expensive medicines. We could get into whole conversations why those medicines are so expensive. That's not, I think, your question. I think what is powerful, if we go back to Ania's talk and some of the aspects I talked about, in that the cost of technologies like sequencing are falling dramatically. So if we can understand better the complexities of cancer, we can choose treatments that are still quite cheap in lower income countries. We've still got that horrible curve I showed earlier on. Lots of data, lots of challenges, but I think it will be relevant, particularly to the worldwide number of individuals with cancer. And it's worth saying that despite our best efforts, cervical cancer is third biggest cancer killer of women worldwide. So those sort of diseases still need medicines that are relatively cheap, but we need to be able to develop genomic technologies that make it easier and also prevent it.
And I think also there's huge scope for better use of data we have already. The kind of common clinical data that's already available before we go down the route of super duper expensive, sequencing assays and things like that. And I think if you look at, so for example, there's some dried blood spot work going. If you look at the circulating DNA technology that's going on, there's lots of work coming out of Cambridge by able to do those kind of sequencing techniques from dried blood spots, which means you can have your blood spot taken wherever with your standard, lancet, stick it on a card and stick it in the post. So, there's lots of work that needs to be done around that kind of accessibility to basic techniques before you go anywhere near some of the fancy stuff we do.
We absolutely need more diversity in terms of the genomes that we sequence around the world. I think it's definitely is pinned down to the economy of basically the places. But as Sarah was saying, if we have more simple tests how you can sample, then that could be sequenced elsewhere. Or there is more technologies that you can take into the field as well. So you could easily basically start sequencing elsewhere. Saying that, you still
environment. So for cancer, it's the reason why microenvironment is so important, mainly due to the nervous system's influence on tumor. For example, GABA immediate proliferation and immune evasion. The reason why microenvironment is so important mainly due to the nervous system.
Yeah, so let me just step back a bit for a second so we're all on the same page. So the way, to cure patients from cancer, you need two things. One, you need to kill cancer cells. And largely, the curable cancers that were cured up until a decade ago were cured because they were very, very sensitive to treatment. So male testicular cancer, for instance, or lymphoma. Even using conventional treatments, you can cure those cancers because you can kill so many of the cells in the body because of sensitivity. However, the really other important thing you need is an immune system that can fight the cancer and keep it under control. And that's the main story about melanoma and why melanoma is now a curable disease. I think your question relates to something very specific, which is how our tumor microenvironment may actually be, and sorry, when we talk about tumor microenvironment, that's what we're talking about, with the cancer cells, the immune populations, and how they're interacting. But there's also evidence now that some of those immune responses are determined by neural tissue, which you can find within the tumor microenvironment. And that's a very new discovery, and the meaning of that is not entirely clear. But there are, of course, experiments in which if you remove neural tissue by genetic or other means, you improve the outcomes in these models of cancer. Anyway, I think it's a much longer conversation than we can have here, but happy to talk afterwards.
Thank you.
Thanks for the panel discussion. James, you mentioned earlier that some of these cells are shed filter up, essentially filter up in the body. Do they also enter the bloodstream or other fluid systems in the body that could be detected? I mean, there are these blood tests nowadays where they're looking for multiple cancers. I suspect because of the fragmented DNA that you have there, that might make it more difficult to detect.
So lots of interesting points there. So I was talking about ovarian cancer and trying to explain why it's always advanced, or pretty much 80% of patients have advanced disease, and that's because you're shedding cancer cells from the fallopian tube into a closed space. They can circulate around, and largely they implant upon the surface of our abdomen, so the peritoneum, bowel, etc. Ovarian cancer cells do sometimes go into the lymph system, and you do see patients who've had spread to the lungs and to the lymph nodes. I think your second point is, so not all cancers behave in that way. So for instance, some cancers like small cell lung cancer are very, very, very good at getting into the bloodstream very, very quickly. And you can detect those cancer cells, certainly in patients with moderate to advanced disease. And in fact, colleagues and researchers at Manchester have been able to culture those cells from blood samples and use them to make preclinical models by growing those cancer cells in rodents. So that's an amazing thing. And then I think the third point you were getting, so we can definitely detect cells in the blood in some cancer types, prostate cancer, lung cancer, but we can also detect DNA that comes from cancer cells that are dying or breaking down their tissues anywhere in the body. And that study is called circulating tumor DNA, and we showed this in 2011 could be done in ovarian cancer because we could leverage or utilize this sort of very screwed up barcoding of the DNA to pick that up in blood. Now, the problem is not all patients have the same propensity for releasing their cancer, releasing DNA into the blood, and there may be relationships with how cancer is responding and other factors. But these non-invasive tests, so instead of having to have a biopsy into your liver or into your omentum, being able to monitor patients over time, I think is still going to be a very promising area where we take those DNA tests. And just going back to your earlier question, DNA sequencing is getting very, very cheap. This is a very equitable technology. Cost of medicines is just going in the opposite direction. So we need to use technologies like DNA and some of the more expensive technologies here to understand how we bring cheap tests to everybody.
These days, actually, there are technologies as well that you could see where that cell comes from. So if you look at the methylation status of the cells that are in the bloodstream, you could actually tell, oh, it comes from lung or it comes from liver. So it's to that degree.
Hi. Just to give some context, I work as a translational researcher for experimental clinics in regulations that allow to do all sorts of tests and all sorts of medicine. And we do extensive genetic testing, and we match all sorts of substances to the tumour type. And what we see is maybe the end game of what you're looking for, which is extreme diversity of the tumour during treatment at the end, and then an extreme change in the diversity of medicines you need at the end. to match that heterogeneity that's been caused through treatments. Practically, do you think that ever we can manage that complexity?
Yeah, so thank you everyone for these great questions, because it keeps us going. Some of us are quite tired, so this is really great. So the main idea here is that as cancers evolve over time, so you start treatment, you only treat a proportion of the cancer cells, the patient's cancer recurs, evolves over time. How do you treat those patients? And that is a very challenging problem. And I think it's also fair to say that probably a lot of the changes are not genetic. So it's not actually just that the genome is changing, although that is a portion of it. There's also reprogramming. So cells are changing their metabolism, in other words, the things they rely on. So traditionally, clinical trials for new medicines are done in patients with recurrent disease who've often had many lines of therapy. And we've refined that slightly by doing the sort of things, possibly not at the same complexity that you're doing, of looking at barcodes and other markers. Where I think the field is really going to have to go is frontline treatment. The reason I showed the neoadjuvant pictures earlier on is that we need to cure patients at the start of their treatment, so we don't give this sort of Darwinian opportunity for these cancer cells to evolve. And secondly, as cancer researchers and cancer professionals, is we have to start using and thinking about using oncology medicines to prevent cancer. So if I think about, I'm not sure I can go back to my fallopian tube picture, but individuals with a BRCA1 and BRCA2 mutation have a lifelong risk of getting breast and ovarian cancer, as well as pancreatic, and in males, prostate cancer. So why can't the medicines that I use in the clinic with patients with advanced disease be used to prevent cancers in individuals who carry faulty genes? Now, there are lots of challenges there: safety, efficacy. Do they work? Do patients want this? Would you rather have intermittent treatment with a low dose of a medicine than having your fallopian tubes removed? So those are complex things. But I think the future of many problems is actually thinking about prevention, because as you're highlighting, I think those scientific and drug problems might be a little bit easier. And if you have a faulty gene, so the other disease I treat, of course, is endometrial cancer, womb cancer, and there are individuals with Lynch syndrome who are at a very high risk of developing cancers for the whole of their lives because of changes, faulty genes they've inherited. So are there ways that we can prevent cancers occurring in multiple areas? Again, thinking about immune responses, etc. So great question, thank you.
So this is Ania, and you mentioned that we are having this huge sort of data bank of data, all these multidimensional data from the cell type to the sort of molecular signaling. Are we focusing on the tumor microenvironment, or is it possible for us to digest that data on a bigger scale, like predicting how the tumor is going to move, or predicting like the immune sort of nature of the patient to see if it's possible for them to withstand chemotherapy, or is it any better sort of therapy? How can we get patients to scan for these tumors earlier so that we can predict that? Because like as Jim said, a lot of the ovarian cancer are at late stage because people don't get scanned quickly enough because the symptom is shown enough. Is that like a problem? Because you know how a few years ago when female present with pain during their periods, doctor just turned them away, but it turns out that's endometriosis and it's very often overlooked. So are we looking at a bias problem? Are we looking at a patient unwilling to go to the clinic problem? Or is there a way that we can use AI and data to predict? It's actually probably that's more for the talk that was before us. Like that was at 6 p.m. with Rebecca Fitzgerald and Mariah Crispin. They gave a very good answers to that. But I'll just say that we focus on the cancer, and what we showed and what I showed in that multidimensional, basically data integration, and that spatial information that Claire was also showing. In a context of cancer, but if you think about context of pre-cancer lesions, then you can step back and you can start to understand how that cancer is formulating. And it does take, as long as I remember, at least like 10 years on average to actually develop the cancer from the time when it actually occurred to the time it's diagnosed. So you have quite of a window of opportunity what to do in that time. So that early cancer detection and early cancer prediction is where, yeah, all those tools I think will help us a bit as well.
Just 2 seconds. So I think women's health has been underfunded, and you're right to highlight endometriosis and other conditions. The problem with ovarian cancer is not about a lack of imaging. I think it's, we just can't image it. You would have to, if you think about it, a screening program, the tests aren't very good. You'd have to screen all women over 50, and so you'd screen millions of women with little benefit and possible harm. So that's one thing. The second thing is, I think AI will be something, I think, that will be important in terms of people working out their symptoms. And so clearly, you know, the most important computer we have, whether we like it or not, is a phone. And, you know, I can see apps sitting on the phone. Obviously now there's apps to track your reproductive cycle, menstrual cycle, fertility. But actually, you know, what about an app that says tummy pain? So if I'm 55 and I've just finished my menopausal transition and I'm getting funny abdominal symptoms, why am I not using an app that looks at me, looks at my reproductive history, knows other things about me, that says, go and get a CA125? Those are the sort of practical tests that I think will come.
And also, the challenge of building apps like that that can provide that kind of information is you really want data sets of at least 10 years in length, if you can have them. So that means finding people who are interested in participating in this kind of research, identifying them, taking their data, getting all that data at regular time points when you don't even know necessarily what data you want to build that successful model. And that's a massive logistical challenge. And also, if you look at, you know, the size of the data sets we're generating these patients, they cost an absolute fortune to store. And it's quite difficult to maintain those data sets and keep them, you know, keep them safe and keep them usable over the times you need to build those kind of models.
Participation in research will help as well, because then it means that you take all those images during the, like, some, I don't know, 10 years, 20 years, and then you build that data set. And once that we have that huge data set, that then you can use basically to build the tools to predict, and detect cancer earlier.
We had Catherine Green here from the chemistry department talking about the COVID vaccine story. This inspired me to ask, she mentioned vaccines against cancers. Is that something that, you know, because you were just mentioning we need to treat the recurrence or prevent the cancer in the first place, but also maybe treat cancer after you've had therapy and so on. Are vaccines a potential solution in this area as well?
Yes, and I think those trials are really starting, again, based around whole genome sequencing becoming cheaper and cheaper, and the knowledge and experience we've had with RNA-based vaccines from COVID-19. So the ability to identify and predict changes in your DNA that causes abnormal protein to be made in your cells, which your immune system can recognize. So I think it's a promising approach. I think we need to really see the randomized trials and the early phase trials with benefit. But again, could be, you know, just again, thinking about cost. Making RNA, making a vaccine is a lot cheaper than making a modified antibody-directed conjugate that can then be used in advanced cancer. So yeah, I think these things are very important and very exciting. More trials needed.
Thank you for the panel. So I wanted to ask you about the triggers of the evolution. Do we know the triggers of the evolution? Do we know some types of the cancer that will that we know that it will evolve further? And can we say that sometimes chemotherapy can cause evolution more, like we said about the vaccines? So vaccines maybe changed some gene sequencing sometimes, and when we're applying chemotherapy, it could cause even further changing and mutations that could cause more evolution. So how can we make the our immune system to work on us and not against us. How to make all of these things more predictable rather than unexpected.
That's a big question that I'm unqualified to answer, so I'm going to hand it over to the panel.
So triggers. So I think there were several questions there. You were asking what triggers were. So, firstly, I think one message which I think is really important, and a lot of this work has come from Cambridge, particularly from Mike Stratton and colleagues at the Sanger, is we have to think about cancer as being an acquisition of changes in the DNA of our normal cells over life. I mean, that's kind of self-evident, but what I'm saying here is our exposures, our lifestyle, how old we are, our genetics, they control how our DNA changes over time. So if you look at the endometrium, the lining of the womb, the uterus, over life through, an average of 30, 40 menstrual cycles, possibly more now if you think about early age of menarche, the lining of the uterus reconstitutes itself many, many, many times. And what happens is you get clonal populations that acquire mutations. So that is a component. I think I haven't forgotten to multiply by 12 here, so sorry about my earlier miscalculation in numbers of menstrual cycles. Those numbers of cell divisions lead to clonal populations that are the precursors of cancer. Same thing happens in our skin, on our bowel, etc. So the idea of triggers, I think, is quite an interesting one because there clearly are factors that change our risk, smoking, alcohol, etc. I don't believe that major life events and stress change significantly our risk, but there is no question that things like immunosuppression, so changing our immune system, so let's say I'm diagnosed with an autoimmune disorder and I have to start taking immune suppressing agents, my risk of viral-induced cancers, particularly HPV cancers, goes up dramatically. So those are really important. I think your question about how we can direct the immune system against our cancers, or at least against our early cancers, that's going to be difficult. But I think in diseases like Lynch syndrome, where you've got this kind of very high risk in multi-organ systems, thinking about vaccines, thinking about molecules that augment recognition of cancer cells, these are going to be very important. Were those the questions you were asking, or did I miss something? Thank you.
Hi. Just on the acceptability of treatments, my last project was trying to prevent and cure cancer by drinking beer, so it's not all bad. But no, hops are great. It's probably a question for Sarah that in your sort of slide, you had the kind of wiggly line from research to clinical practice. Where do you see the big pain points? So, and obviously money always, but is there a sort of kind of regulatory thing? Is it things aren't quite ready in the science yet to go from that sort of complicated down to the quick tests? Yeah. Where is it?
So I would say we are not sufficiently skilled in evidence generation for these kind of methods. So if you have a new drug, there's an extremely clear path towards understanding how that works. Everybody knows how you would set up the appropriate clinical trial infrastructure to do that. But I think if you look at data-driven methods, I don't have a digital clinical trials unit, right? I can't go to CCTC and say, hey, look, I've got a new algorithm. Can you sort me out a clinical trial? And also on top of that, I think if you look at a lot of the data validation activities that need to happen around data types you want to build an algorithm around, it's extremely challenging. So this has been done in Cambridge. So one of our colleagues, Raj Jena, has a method for automatic image segmentation. That software is a medical device. It's used for radiotherapy planning. And I think we are not yet skilled enough and adept enough at dealing with data-driven health interventions on this kind of level. And on top of that, there are the kind of infrastructure problems. So around compute, around having a digitally savvy healthcare workforce as well that understands how to integrate these technologies in the clinical setting. Because if you look at how far technologies come into our everyday lives, I think in many clinical settings, we're still very much behind the curve in that. And I think the digital environment in the clinic is out of step with the rapidity of development you see in every other aspect of our lives. And a lot of that is regulation, and a lot of it is cautiousness on behalf of a number of bodies, not least because everyone's terrified about data leaks or things like that as well.
And so other issues, I mean, I guess you touched on it, maybe sort of things like standards for imaging, or, you know, sort of, certainly in my world, you sort of get a lot of problems with, you know, the machine from one manufacturer does things a certain way, but it's not compatible with.
So I would say that actually medical imaging is quite a mature field for dealing with that, simply because it's been forced to be digitally native for much longer than pretty much almost any other aspect of medical data generation. I think, so if I look at another project I'm involved in, so increasingly for clinical trials, you have the sponsor, for example, a pharma company that wants to collect data, has a list of data it wants. It might want to know your hemoglobin levels, your blood pressure, your blah, blah, blah. Most of that is available in electronic formats, and yet it's incredibly difficult to reliably extract that information from a range of clinical systems across all the different sites you're running that trial, and for the sponsor to have confidence that because they've asked one site for hemoglobin, that they're getting the same thing back from all the sites. So it's a real logistical problem that's not particularly sexy to solve, right? It's not going to win anyone a Nobel Prize, but that's the kind of cost of progress of dealing with all those kind of digital technologies, is because there's a huge amount of heterogeneity in the hospital landscape. And I think it's really interesting because the NHS is seen as a centralised body, and yet it's not. The NHS is not one monolithic organisation, and it's a real challenge.
Thank you. You mentioned the increasing costs of drugs, and when you truly analyse what the immune system needs to work against cancer, you see a phenomenal range of drugs required simultaneously. So checkpoints alone, there's at least 10 identified. Are we looking at one point to be using 10 different checkpoints? So that the direction that we're going is going to get worse when you really truly understand how the immune system works in terms of the number of drugs required to really work against cancer. But at the same time, there are lots of off-label drugs which are free, which are showing very good evidence when combined with immunotherapies. So, for example, ivermectin in animal studies is making tumours immunogenic in breast cancer. Antihistamines and statins are doubling outcomes with checkpoints. Do you think there's a mechanism to get these drugs into clinical practice? Because primarily the trials are funded by pharma.
So now we're back to the cost of medicine. So first of all, I completely agree. Complexity is at the heart of all biology, particularly the immune system. And so there are multiple targets and multiple ways of interacting with the immune cells that are responsible for curing cancer. So largely that's around T cells, adaptive T cell responses, but there are also roles from B cells producing antibodies. So I think the big thing here is where's the biology and where's the compelling preclinical evidence. And we have been working for some years on repurposing medicines, and I think, you know, taking medicines that have already been shown to be safe in humans, perhaps not for cancer indication, obviously, be able to use them in combination, is really important. That also means we need enough charity and other source funding to do the trials, because pharma will not fund those trials for reasons that are evident about, you know, selling product. And, you know, I'm not here to bash pharma, but I think there are clear business objectives, and that's about selling the assets you have or acquiring other people's assets so you can sell them. It's not about using cheap medicines in combination. So the one thing that I think is important is that over the past 10 years, the number of investigator-led phase 3 trials has dramatically fallen. So for everybody else's benefit, that's, you know, strong evidence-based trials that are really testing whether a medicine works in the clinic. Those have been largely replaced by pharma trials, and we need to reverse that. But that's a multifactorial problem all the way back to CRUK, MRC, other charities funding innovative trials and funding innovative research. And we need new ways of bringing people into cancer to do that dimensionality drop. You know, how do we take huge amounts of information, turn it into the key things, and that's about mechanism.
So I have a comment on that. So I think, you know, you do have this idea that pharma are driving up the cost of medicines, but I think the opposing force of that are healthcare systems, which are absolutely motivated to find cheaper, better, more effective treatments. Do they have the might of pharma? No. But are they important in moving, you know, in kind of providing a bit of tension back to others? Yes. And I do think that the methods that are coming out of the labs give us the opportunity to know that if it is a very, very expensive treatment, we're only going to deploy it whenever we know it's going to work.
So that's it. I think we're probably done for time. Thank you all for coming. Thank you to the amazing panel. Also, I think someone was asking earlier, but this has been recorded, so if people are interested in the slides, that will be on the recording, which will be on the festival website later in the month. There's also an exit that way, which has a sort of art installation of black scientist portraits, which has amazing portraits, but also little bios about the scientists. If people are interested in passing that, you might have seen some of the photos on your way in. But yes, thank you to the amazing panel once again.
Thank you very much.